All right, time to get started. Good afternoon, everyone. Welcome to the vaccine session of our first virtual New England Science Bootcamp for Librarians. I'm Melanie Radik, and I will be hosting this session. In order to ensure a respectful and inclusive environment, please note our code of conduct on the New England Science Bootcamp website. The link is in the chat box for your convenience. This session has live closed captioning. It's down in the multimedia viewer box below the chat box. If you experience any technical difficulties, please message all panelists in the chat box. Our speaker for this session is Dr. Wilmore Webley, Associate Professor of Microbiology and the Director of PreMed and Pre-Health Advising at the University of Massachusetts Amherst. Dr. Webley earned his master's and doctorate in microbiology right here at UMass Amherst, with expertise in immunology, infectious disease, host pathogen interaction, and concentration in vaccine development. Among many other rewards and honors, he is a Fulbright Scholar, a recipient of the Manning Prize for Excellence in Teaching, and serves on the editorial boards of the International Journal of Vaccines and Immunization, the Journal of Medical Microbiology and Diagnosis, and the Journal of Microbiology and Experimentation. His research focuses on infectious disease mechanisms and the role of individual infections in chronic diseases. Specifically, the Webley Lab is pioneering work in chlamydia vaccine development and the role of pathogenic microbes in asthma initiation and exacerbation. All attendee microphones will be muted for the duration of the presentation, so we ask that you enter any questions you have into the chat box and make sure to send it to all panelists. Our Q&A moderators will ask as many questions as we have time for at the end of the presentation. Now I will turn the session over to Dr. Webley.
Thank you very much, Melanie. Waiting for the ball to share my screen. Okay. Thank you very much. So hopefully everybody is seeing that shared screen in full screen mode. Thank you. Good afternoon, everybody, and I thank the organizers for this great boot camp that I've had the opportunity to attend in years past. And this great experiment has worked out really well. The first presentation was really, really good, and I thoroughly enjoyed it. So basically what I'm going to do, I'm going to talk you a little bit through some of the processes that we work through in my lab for vaccination, and then pivot to COVID-19 and SARS-CoV-2 vaccines, and hopefully some of the principles that we'll learn along the way in chlamydia vaccine development will actually help you in understanding that second part of this. So I love this statement here from the World Health Organization that says that two public health interventions have actually had the greatest impact on the world's health, and those are clean water and vaccines. And that has never been truer than today. And essentially, vaccines have been around for a long time, and although we have attributed the founder of vaccines to be Edward Jenner, seen here in this picture, scratching the hand of young 13-year-old James, 8-year-old James, to give him cowpox so that he could be immunized against the smallpox. We know that vaccines had been around long before that. Buddhist monks, for example, were great at taking venom from snake and inoculate themselves with it so that they could develop antibodies to or anti-venom antibodies overall. So vaccines have been around for quite some time. And so now I just wanted to show you kind of the timeline here for vaccine development, starting with 1796, which was when Jenner introduced the smallpox vaccines. And from there, you can see that Louis Pasteur continued. And as you continue through the years, the decades, the centuries, what I want you to notice is that there's no huge cluster of vaccination. There's no period where you have just tons of vaccines created compared to another period, so to speak. And that's kind of interesting. And it underlies kind of this mysterious immunology of how vaccines work, because essentially when Jenner started out, what he essentially did was use a crude vaccine extract to do this. In fact, if he was to try that today, he would have been locked up in jail because that's just not the way you do it. He didn't have an IRB. He just kind of just used the first person he could get a hold of. But vaccines have evolved over time so that we reach to this place now where we see that vaccines have eradicated several diseases, including the dreaded smallpox, and has significantly reduced the incidence and prevalence rates of certain infections, especially those that have been detrimental and deleterious to human beings. It turns out that the science of vaccination actually predated the science of immunology, which to a great extent, immunology up until recently was actually adaptive immunity, which is the basis for immunology and vaccination. And so here I'm going to introduce a little bit of basic immunology. And you hear people talking a lot about making a vaccine and how vaccines are made. But it starts fundamentally at the molecular level, where what you're asking yourself is, which proteins are important to the pathogen that it actually needs to carry out its developmental cycle, that it needs for living, that we can actually target and block so that that pathogen won't cause disease. And so we get into the realm of basic immunology of antigens and antibodies. And antigens are essentially any entity that comes into the body that is foreign. In fact, any protein could be an antigen. So could probably any carbohydrate from the food that we eat to anything that we encounter in the environment. These can all be considered to be antigens. And so beyond the antigen, you have what are called antigenic determinant sites. And these are parts of the proteins that are specifically recognized by the immune system. So the immune system doesn't just recognize a whole bacterium as a single antigen. It actually recognizes it as a plethora of antigens for each stretch of maybe 5 to 7 amino acids, depending on how the protein folds, and you could actually have an antigen. And antigens can be linear. Your epitopes could be linear, or they could be conformational, just based on the curvature and the 3D or 4-dimensional folding of those proteins. So what you're seeing here is that you're seeing this antigenic determinant site, or epitope, colored here in orange. And you're seeing this antibody made by B cell 1, because your B cells are created specific to a particular antigen, and the receptor on the surface of your B cell is an antibody, an IgM antibody. And that now circulates, that B cell circulates, and when it finds an antigenic determinant site that matches, like a lock and key, as was stated this morning, that site on the antigen, then this B cell now is triggered to clonally expand and make more of itself. And that's how you actually get an antibody response, because then these B cells will differentiate after they replicate and expand. They'll differentiate into plasma cells, and it's the plasma cells that will specifically make antibodies that will now come into the circulation and bind. And that B cell is very different from your B cell 2, which is binding to antigen 2 here, and you could go up to as many as you can think of, from the surface to whatever is secreted in these proteins. So the basic immunology that governs vaccines is that you have some antigen or antigenic entity like the coronavirus that we're going to talk about in a little bit. And as was stated this morning, it has these spike proteins on its surface, but these are not the only antigens that it has. The structural proteins that it has for its capsid, even its polymerase proteins, all of those are targets for the body to make an antibody response against. So this virus entered, and you saw a great explanation of its entry this morning. It's taken up, it has an RNA plus strand RNA. And it comes in, and because it's an RNA virus, a positive strand RNA virus, it doesn't need to go into the nucleus. It sits in the cytoplasm and uses the ribosomal proteins of the host to make what proteins it needs and to replicate its RNA. And essentially, as it's doing that, each of these proteins that is being produced in the body is loaded onto what we call MHC, major histocompatibility complex proteins, and they present these on the surfaces of the cell, even as the cell is being infected and being taken over, its machinery is being taken over. And so now what happens is that you have what is called antigen presentation by cells like macrophages and dendritic cells. These are professional antigen presenting cells, and these now get presented to T cells, and these T cells offer help to these original B cells that had encountered the antigen, as well as to any differentiated T cells that you have. And it's the cytokines and the chemical messengers that are produced by this helper T cell that actually gives the fuel to these B cells to proliferate and differentiate and make the antibodies that you need, so that these antibodies now can now go out into the circulation, find any of the original viruses coming in, whether they're processed or not, bind to them, and hopefully have antibodies that are specific to binding to the spike proteins over here. And that will prevent the virus from actually attaching to its receptor, the ACE2 receptor, to get in. Of course, it has other structural and non-structural proteins that antibodies are also going to be made against. And hopefully those antibodies might induce complement or some other parts of the innate immune response that would lead to the destruction of these viral particles, in addition to the destruction that you would get from the cytotoxic T cells that are produced. What this diagram does not show you in great detail, and just down here, is that what you have after that are what we call long-lived memory B and T cells, and that is the basis for vaccination, that you have these circulating antibodies, but you also have these memory cells. Because this response that I just showed you takes about a couple of weeks before it reaches peak, and I'll talk more about that later. And what you want in order to protect yourself from most pathogens is that you need actually a shorter response. Because eventually what you want to get to is what you've heard probably in the news and you've read about, what we call herd immunity. And it's the principle that if you have a lot of non-immunized individuals and you throw a sick person or two in the mix with a contagious virus or bacterium, then what will happen is that in a very short time, most people will become infected. But the more immunized or protected people you have, either they're immunized to acquiring the infection naturally or they're immunized to getting a vaccine, eventually you have enough people in the population who are protected from the virus. And this is an animation showing you when you have no persons who are vaccinated versus when you have over 90% of people vaccinated for just about any contagious pathogen. And as the animation is showing you, what essentially happens is that you actually have a greater level of protection coming through from those individuals. Here's the problem with all of that. We've never seen a situation where you have developed herd immunity, where we have any population with herd immunity without vaccines. And vaccines take a long time to make. And so while we're talking about this accelerated vaccine process, if you remember, the typhoid was around for 105 years before we had a good vaccine. We still don't have a strong vaccine for malaria. And as you can see, there's several pathogens, including HIV, that have had trillions of dollars invested in vaccine development that still doesn't have a vaccine. And that is also true for chlamydia. So let me tell you a little bit about chlamydia. Chlamydia is the bacterium that causes sexually transmitted chlamydial infections. It is the most common bacterial sexually transmitted disease worldwide. It accounts for about 127 million new cases worldwide each year, about 1.8 million of those cases in the US. And in the US, that's a 19% increase since 2014. Now, what does it cause? It causes what we call pelvic inflammatory disease, which is a progressive inflammatory condition of the genital tract that ascends from the cervix all the way up to the fallopian tubes and into the ovaries. And it can lead to ectopic pregnancy, where the pregnancy takes place in the fallopian tubes. and also infertility in both males and females. The problem with chlamydia is that 70% of the cases of chlamydia trachomatis infection are asymptomatic, and so you might not even realize that you have this infection that's leading to these types of issues. It's also the leading cause of preventable blindness worldwide, a condition that we call trachoma, and you can see the map here of the endemic areas for trachoma. And a lot of this is because of the areas in which you find this, where it's low hygiene, the flies are able to carry it around. Chlamydia has an interesting developmental cycle that is being portrayed here. It has a biphasic developmental cycle where it enters the cell as this spore-like elementary body that is infectious but not replicative. And then once it gets in, it transforms into a little bit larger reticulate body that is replicative but not infectious. And so what you have is that the infectious EB comes in, and what you're seeing here is a little pit, this vacuole that gets taken in into the cell through endocytosis. And over time, it forms this larger vacuole that replicates, where the microbe replicates by binary fission, until you get anywhere from 300 to 1,000 of these replicative reticulate bodies. It transforms itself back into the infectious form, just differentiate into it. And this is an immunostain showing you the sizes that these can get to. Here you can see just a regular microscopy. And eventually what happens is that it pops open after about 36 to 48 hours, and it releases these progeny into the circulation. You can see how the lumen of this inclusion that it forms just fills up. And if you look carefully, you'll see when it just spews out all of these new elementary bodies, and these infect all of the cells around. And so because of its asymptomatic nature, and because of its biphasic developmental cycle, where the infectious form, there's no way of genetically manipulating it, we are looking to the fact that if you don't have a vaccine for chlamydia, it'll be very hard to slow down or stop the spread of this disease. And so when one thinks about vaccination, the antigen that you're going to use as your immunogen, that you're going to immunize with, comes to the forefront. That antigen is really, really important. And in the process, before you reach manufacturing, all of these areas from storage to formulation is the manufacturing process, and that has to be scaled up. But even before you get there, the question is, how do you deliver whatever antigen you're going to use, whether it's going to be DNA, RNA, protein, carbohydrate? You don't just put this into an individual and they make, or an animal, and they make antibodies against it. It has to be presented in a certain way. In other words, your immune system doesn't care much for a buffet. It needs a sit-down dinner. It needs a good presentation. Without that presentation, you don't have a good immune response. And so you typically need some system to do that. And so the main objective of our research was to find a presentation system for a vaccine antigen that was human compatible, that over the years of testing, we found out that no single antigen on the surface or that has been secreted by chlamydia could work. So we need something that would give us a multi-subunit platform. And we needed to find promising vaccine targets, ones that the pathogen needed. Those sitting on the surface, like the major outer membrane protein or the outer membrane complex that it needs for attachment and entry. Those that it needs for this differentiation step when it's coming from the elementary body to the reticulate body. So essentially, we're targeting those things. And these are the criteria that we looked at. Is it surface accessible? Is it conserved among the different types? Because chlamydia has about 14 different types that we need to cover in a vaccine. And is the antigen, is the protein that we're looking at essential for the survival of the virus? And we can't make it something that the body already has and uses, otherwise it will bind to that and cause problems in the body. And so that leads us to what we call a delivery system for a vaccine. And we sought after a nanoparticle. And you could create nanoparticles mechanically, but we looked for a naturally produced nanoparticle. And that led us to Halobacterium salinarium. And the name is a misnomer because it's really an archaea and not a bacterium. But suffice it to say, it's a haloarchaea. It loves salt areas and salinarium. And this is actually one of these solar salterns. If you're flying the airplane over certain areas in the Midwest, you'll see these different colorations. And these are actually your haloarchaea. These are these archaea that live in these salt water areas. If you go to Salt Lake City, for example, in the Great Salt Lake, you'll find a lot of these microbes. And essentially, here's one in the San Francisco Bay area if you go there in the evening. So if you go to these areas, very high salt concentration, you allow the water to evaporate, and the salt crystals will look like this. They will have this tinge of pink in them, and that actually tells you that this halobacterium is actually living inside of them. If you crack open these salt crystals, you'll actually find it in there. So it turns out that the genome of this unique archaea was actually sequenced at UMass Amherst by Shilda Sommer many, many years ago. And when he sequenced it, he realized that in addition to its 2 mega base pair chromosome that it uses for its function, it had 2 extra chromosomal plasmids. This larger plasmid and then this smaller PNRC100 plasmid. And this small plasmid had on it all of the genes, the 14 genes that were responsible for making these nanoparticles inside of the bug. You might say, why would this bug make these nanoparticles? And each of these haloarchaea that you see here make about 70 to 90 of these nanoparticles, all of them the same size, football shaped. It turns out that it makes these nanoparticles so because they're gas permeable, and it actually helps the pathogen, well, not the pathogen because haloarchaea is not pathogenic, it actually lets the microbe to float up and down the water column. And these look like this. They look like a football. And they allow gases in. The microbe controls the gas that's going in and that's being left out. And when the sun is out and it doesn't want its DNA to be damaged, it releases the gases. It takes in these gases and it gets down to the bottom of the lake or ocean that you find it in. And when the sun is not too hot, it floats to the surface. It turns out that we consume these all the time. Here's a salt crystal that has been cracked open and you find it in there. If you're into soy sauces, they're preserved with this salt, and you actually have them in there. If you use solar salt as your salt alternative, or if you're into beef jerky and beef jerky underwear, these actually are loaded with haloarchaea. You can actually crack open these salt crystals and grow it up. So we actually went in and looked at the entire plasmid that actually had these surface proteins coded for it to make this gas vesicle. And we engineered a site into the outermost protein called GVPC. And this is actually the protein that actually ties this structure together. Without it, the structure is not very stable. And so we inserted our protein genes of interest that we think we want to make a vaccine against into this GVPC gene, and the promoter reads through it. And so essentially what it does is that it produces a chimeric protein, the gas vesicle outer surface protein, and then our protein of interest on these wrappings around the gas vesicle itself. And the truth is that we don't really know what it looks like on the surface, but what we suspect is that it has multiple copies of our gene of interest in there. And we've used multiple genes to do this. And so what you're seeing here are gels that we've used different serobars, the ocular ones and the genital tract ones, and we put in different proteins, from the major outer membrane protein to the polymorphic outer membrane proteins, the ONCB proteins, because we're just not sure which ones of these are going to generate the strongest immune response. And what I'm showing you is that you actually have an empty plasmid that doesn't have anything in there, and then you have an increase in the size of the plasmid as and this is the plasmid here. Here is the engineered site right in the GVPC. What you're seeing is that you have an increasing sizes when you have no insert versus multiple different sizes of inserts. And so what we've demonstrated is that this plasmid will express these inserts. And then the question is, will it actually produce these recombinant gas vesicle nanoparticles? And for those of you who know molecular genetics, we actually spheroplast these gas vesicles, these haloarchaea, we have a strain called ST109 that doesn't produce any of the gas vesicles, and then we have a strain that does. We strip off the outer membrane, allow it to take up this plasmid that I just showed you, and it turns out that when it takes up this plasmid, here we're growing it up, it will produce colonies. If it's producing gas vesicles, produce pink colonies, and if it's not producing those, then it will be red colonies. And so we pick the pink colonies because those are the ones that we want. It has our plasmid-laden haloarchaea in there, and we grow those up. And when we do that, we can now scrape these off the surface of the massive plates. It's growing in high salt concentration. It's a slow grower. But we can purify them until we get these purified gas vesicles. And what we realized is that once we started growing them up, we could actually put them in vats on the windowsill, and they just grow up. These are the different boxes with the different inserts. And over time, what we have, you can allow the media to evaporate, and you have these salt crystals. And inside of these salt crystals are recombinant gas vesicles that have chlamydia proteins on their surfaces. And all we have to do is essentially to throw these archaea into water, because it grows in such high concentration of salt. So we throw them in water, they just pop open and they release the gas vesicles that we can purify them out. So essentially, what we're thinking that we could create is actually a vaccine in a salt shaker, where we could actually crystallize the salt and people could use it. And then they, as they're getting their salt, they would actually get the antigen in there through the archaea, since it doesn't grow in humans, doesn't cause any problems where disease is concerned, and we could actually get inoculated that way against the microbe. So, sorry, my PowerPoint just got stuck here. Yep, there we go. So in order to demonstrate that we actually have proteins that we're expressing, this is a Western blot, and we have antibodies against the gas vesicle surface protein. And these are the recombinant ones that we've created. And what we do is that we run them on a gel, we transfer them onto a blot, and we stain and we ask whether or not they're actually expressing the gas vesicle protein, the one that we interrupted. And the answer to that is yes. We're seeing different sizes of it because of the nature of the gel. And then we ask whether or not we're finding chlamydia proteins on these gas vesicles, and the answer is still yes. And then we took human serum to both chlamydia pneumoniae and chlamydia trachomatis. These are different strains of the chlamydia. Pneumonia is the inhaled one. The C. trachomatis was the genital strain. And we asked whether or not we could find antibodies in these naturally immunized sera from people who have gotten the infection, and if these actually reacted with the gas vesicles, and the answer is yes. So essentially what we're demonstrating is that this nanoparticle is able to express the chlamydia proteins on its surface in what we call native form, in the form that you would see in the body if that was expressed. And so then the other question now becomes, can we actually use this to immunize somebody with, and could we actually demonstrate that we can actually lead to productive antibody production? Because this is all in tissue culture. We ran it through tissue culture and looked at the production. This is all in vitro studies. So we did a mouse model. These are our mice. We used biopsy mice. And we tested our different constructs of our gas vesicles with recombinant gas vesicle nanoparticles in them. And so essentially what we did is that we're able to immunize these mice subcutaneously with these gas vesicle nanoparticles. And we're demonstrating that we're getting antibodies to specific proteins, but not to others. You see these POM proteins and these regular MOM proteins. When we combined all of them, MOM and POM, we're not seeing a reaction. We're seeing one to this OMCB protein and to the all, and when we realized the all, it's because the OMCB was in it. So it demonstrates we're getting IgA antibodies and we're getting IgG antibodies because IgA antibodies are mucosal antibodies. And since chlamydia infects mucosal surface, we actually need mucosal antibodies. But because it can get into the circulation, we also need IgG antibodies. So then the question becomes, yes, it's producing, the mice are producing antibodies to it, but can it actually protect them against a challenge? So what we did is that we got female mice, and we used chlamydia trachomatis, and we infected them with the chlamydia after vaccinating them and boosting them twice. And after allowing them to sit for 2 weeks, for a month, we went back and we checked to see if the vaccine actually protected them from the type of infertility and tubal damage that we see with chlamydia infections. And what you're seeing here is that we're able to swab the vaginal tract of these mice, and we're able to grow up the chlamydia. And this example is from our unvaccinated group of mice. And then what we do, we actually take the swabs, we grow up the chlamydia, and we count them. We see what is the load that we're seeing from the infected animals versus the uninfected animals, and also the ones infected with different combinations of these recombinant gas vesicles. And what we saw is that here's our control mice that did not get the gas vesicles. Instead, they got the vehicle, which is basic saline. Here is the major outer membrane protein. There was a reduction in genital pathology, reduction in the type of damage that we're seeing, but still not to the level of protection based on the pathology that we have. But here we're having this, again, this construct that was a little bit unique because we actually used what we call immunoinformatics, where we specifically looked for a protein that had both B and T cell epitopes on it. In other words, would induce both a B and T cell response. And that was the one that we were seeing really good response for, although it didn't protect against all pathology and all infections. So here you're seeing our control animal, the genital tract. This is the fallopian tube. So this is the right fallopian tube, and this is the left one. The left one actually popped when we're taking it out. That's why you don't see the fluids in it. But this is the way it's supposed to look. This is the normal fallopian tube, and this was one that actually got the cocktail of the gas vesicle vaccine, protected it from pathology. And here you're seeing the inflammation leading to this full pathology that we're having here. And so essentially what we're able to conclude from this is these gas vesicle nanoparticles could actually be a vehicle for the delivery of a chlamydia antigens, that it could display these chlamydia peptides that were recognized by antibodies in the human system. It had a natural time release system overall, and that overall it induced the production of B cells, T cells that responded to it. We had antibody response. And so it kind of checked off a lot of the marks on this original thing that, yes, it's a nanoparticle and it can deliver the vaccine antigen, that it actually induced the right type of immune responses. What we didn't have, though, is that we didn't have the combinations of antigens to induce the strong enough immune response to be completely protective. And so what we're in the process of doing now is creating more antigens like that one that I showed you that was very much protective. And hopefully we'll be able to get to the place where we have enough of those, or in the right configuration or at the right dosage, where we can actually have a protective response that is able to give you a 60, 70, or 80% at least protection against these this microbe in the animals. And then we'll be able to maybe go to a phase 1 clinical trial overall. So this essentially kind of sets up some of the components and some of the thinking that one has to go into vaccine development with as we think about the current pandemic that we find ourselves in, as I switch gear here. So pandemics have always been with us, from the Antonine plague to the bubonic plague that killed over 200 million people, to the Spanish flu in 1918, 1919. HIV/AIDS is also a pandemic, to this new COVID-19 pandemic that we have recently. We've always had pandemics, and throughout these pandemics, we've learned some lessons from them. Today, we're battling a pandemic like we've never seen before. This is a highly contagious coronavirus that started in Wuhan, China, sometime at the end of November to early December in 2019, and that since then, according to this Johns Hopkins tracker, has actually infected over 7.4 million people, causing the death of over 417,000 And in the US, we just passed a milestone of 2 million people infected with this virus. We're seeing surges in places like Brazil and other parts of South America, as well as in India. So the question is, what is this virus? And our initial speaker did a really good job on showing you this coronavirus. And I'm just going to spend a little bit more time on this. So you might see this cartoon on the CDC site and several other sites that show the coronavirus. But what I'm showing you here is actually an electron micrograph of what it actually looks like, of course, falsely colored. And so what you see here, you see on its outer surface the spike proteins that you learned about this morning. And these spike proteins are what actually give it this name, this crown-like look, because cronum means crown in Latin. And so the disease is called COVID-19, coronavirus disease 19 to 2019, when it was discovered. So this is the disease. And then the virus that causes it is the SARS-CoV-2, and this is the severe acute respiratory syndrome virus 2. And essentially, up until this point, we had known about 6 or so coronaviruses, 4 of which were responsible for just a common cold. But we knew about SARS. This is the original SARS coronavirus that we learned about in 2002, 2003. That caused an outbreak that turned into a pandemic really quickly, but it only infected just over 8,000 people. Importantly, it killed about 744 of these individuals. You're talking about almost 10% death rate. And then in 2012, a cousin of this SARS, MERS, which is a Middle East respiratory syndrome virus, started, and up until this point, it has been confined to the Arabian Peninsula, where it actually gets transmitted from bats, like SARS. In this case, it gets transmitted to camels. And from these Romeric camels, it actually infects humans. And as a result of that, we haven't seen widespread contagion going around the world. But up until this point, we've had 2,500 cases, or a little bit more. Deaths are up to 860. So essentially, you're talking about nearly a 30% infection. And then, of course, this SARS-CoV-2 virus that we had. And so when you look at the family tree of these viruses, as you're seeing, a lot of them cause the common cold in different clades here, from the alpha to the beta coronaviruses. You're seeing that most of them cause the common colds. And these that you're seeing in red here are the ones, the 4 that we knew before. If you notice, they don't really have any fancy names. They just have this HCOV with some alphanumeric character. They are known to cause the common cold, and we never really worried about them until, of course, SARS, which is right over here in this clade, where you see SARS-CoV-2 also sitting. And the original bat coronavirus is sitting on the same branch of this tree. MERS is down here. It's also a beta coronavirus, but more distantly related. And so we know that these coronaviruses, bats are their natural hosts. And these bats are mammals. And so essentially the problem with them is that, like us, they like to have communities that they live in, which means that they transmit much more easily. And that they also fly, so they can go to very far distances and infect other groups of bats. Man have been crouched on bats as well, and so as a result of that, the people live very closely to them, especially there in China, where you see the outbreak happening. They also eat bats, and so as we said before, this virus binds to the ACE2 receptor. It has a co-receptor, the TMPRSS2, but lots of cells have these, and some of the confusion that people have been having about the different symptoms that we're having from this virus is because of the multiplicity of organ systems that it is able to infect, not just the lung, but, as you heard this morning, the heart, because the blood vessels actually have high levels of ACE2. We now know, for example, that we thought that, for example, people who have asthma would be way more susceptible to pathology from this virus. It turns out that people with asthma have a lower level of ACE2 receptor because of the disease. So as a result, they haven't gotten as much infection as would have been expected, especially in places like New York City, where the prevalence of asthma is over 10%, you would have expected more asthmatics to die from this disease. That's not happening because of levels of ACE2 and presumably maybe TMPRSS as well there. So for people who get the disease, most people who become infected have kind of mild to moderate disease, where they might have a little bit of cough, a little bit of congestion, or maybe not. For people who have severe pathology, what happens is that the immune system responds to the virus in a really aggressive way, because initially the virus actually inhibits the innate immune response that we have to viral infections, interferon gamma and type 1 interferons that are supposed to alert the immune system and alert the body to the virus. Those are blocked by the virus. And then once the virus starts replicating, the adaptive immune system catches on. And then you start having what we call a cytokine storm. It starts releasing these pro-inflammatory cytokines, which leads to the buildup of this mucus in the lung, which is what you're seeing depicted here as yellow. And that mucus now, that fluid in the lung, is what causes difficulty in breathing. And so oxygen can't really diffuse through this. And these are typically the people that you put on a ventilator. However, this accounts for less than 6% of people in general who are infected with the virus. And what we see is people with high hypertension, diabetes. We know that people who are hypertensive have a higher level of ACE2 receptor on their surface, so that might actually help to account. And all of this goes into thinking about how do you make a vaccine. The other thing we know about this virus is that it's extremely contagious. And it can sit around on surfaces if it's in fluid for varying periods of time, depending on what surface you have, whether it's aerosols or stainless steel and plastic. And if you don't use some type of disinfectant to kill it, it could hang around for a long time. But let's go back and focus a little bit more on the structure of this virus. This is a rather large virus. This is a virus that has 29,000 base pair bases with about 29 proteins in its structure. This is very large for a virus. So in other words, it has lots of structural proteins and non-structural proteins that it has, including your RNA polymerase and these non-structural proteins that you have on its surface. And these are going to become important as you think about vaccine. Now, we already said that what it uses is these spike proteins to actually bind to the cells. So they are, of course, the most obvious target for vaccine development. And what you're seeing here is the membrane of your SARS-CoV-2, and then you're having this gray part in between it is actually your S protein that would bind, presumably, to your ACE2 receptor. So now you have this animation showing you how you can actually have this opening up of the SARS-CoV-2 protein, and then you actually have the ACE2 binding in this cleft, and that's how the virus is able to actually get in. And what I'm showing you here is that you're having all of these B cell epitopes. Remember, we talked about B cell epitopes right in these binding areas and on the surfaces of this spike protein. And these now will definitely become targets for vaccine development and are indeed the major targets for developing vaccines. Why? Because if you're able to get an antibody that binds in here, it will prevent the ACE2 from actually binding. It will block it. This is what we call a neutralizing antibody, that as it opens up, or even without it opening up, an antibody binds here and here, it prevents the opening up, and so ACE2 can't bind, which means that the virus can't attach to the cell and get in. Once the virus gets in, it induces initially an IgM response, and this is not unique to this virus. We have 5 different types of antibodies, and the first antibody to be produced and detectable is your IgM antibody. After that, you have an increase in IgG, which is long-lived, and which will go out in the circulation, and it is these antibodies now that would be your neutralizing type of antibodies. It is very likely that you might not have an immune response that will be protective after your initial vaccination if you create a vaccine, and you would need a booster. And the reason for that is that in your primary immune response, it takes a long time for you to have a peak level of antibodies, and the height here could be describing antibody response. They're calling it the adaptive immune response here. And so the more robust the immune response, the stronger it is going to be, the more antibodies you're going to have. And for many people, they'll produce a lower level response. It will take a longer time because you have to have the differentiation, the replication of the immune cells. That takes time, and it peaks right about 14 days here, about 2 weeks. However, your secondary response is faster, and that's why vaccines work. So when you introduce the same pathogen again, you now have memory cells, and the memory cells are able to recognize the pathogen. And instead of taking about 14 days to show this peak, it's showing you a peak in 3 to 5 days. And that's how you're able to bind up then all of the microbes or most of the microbes that come in. So here's a fact about vaccines. Vaccines do not protect you from getting infected. What they do is once you're infected, they protect you from experiencing the pathology caused by the virus because they're actively neutralizing the virus. And over time, as you get exposed to the virus, it continues to keep your immune system boosted with these antibodies. So what are the components of a vaccine? A vaccine needs an antigen, and of course it needs something to enhance the immune response, especially if you're using a subunit vaccine. In other words, you're not using the whole particle, you're using proteins from it or RNA from it or some other component that you're using to put into the system. So you do need an immune enhancer that we call an adjuvant overall. Again, it gets picked up, you induce antibody response, and in most of these types of responses, you need an antibody response. Certain pathogens like viruses that infect cells, you need a T-cell response as well because antibodies can't get the virus while it's inside of this. So what we've seen is that we've seen a few animal models pop up. So there's this ACE2 human ACE2 mouse. That's a chimeric mouse that has ACE2 receptors on its human ACE2 receptors on its cells. We've seen ferrets being used, and of course, non-human primates are being used. And essentially what you want, again, is that you want to generate antibodies that will neutralize this virus. So let's say you have passed this preclinical stage, and you're getting now into looking to see if you can actually have development of a vaccine. You have to go through what we call clinical trials. And so the first thing that you do is that you have to decide whether or not you can use antigen A, antigen B, or a combination. In other words, you have to decide what type of vaccinate are you going to utilize, right? And that's going to start what is called your preclinical trial, your human trial, your tissue culture testing of this. And so you can use anything from a weakened form of the virus, an attenuated form of the virus, not desirable because attenuated viruses can reactivate, so people don't really like that. You could use a killed version of the virus. The great thing about the attenuated version of the virus is that it replicates inside of the host without actually causing the disease. So you want that because it will constantly, over a period of time, induce the immune response. If you're using a killed microbe, then that will give you a one-time immunization, then it gets destroyed and taken out of the system, but you can get immunized against it. Again, are you actually killing all of them? You can use subunit viruses, nanoparticles like what I showed you for my vaccine. You can use a DNA vaccine or an RNA vaccine that you can embed. You can use a viral vector like adenoviruses to display. This is the display and delivery component. So after you have demonstrated in an animal model that you can get a response, that the animals are not falling over dead, then you actually go into the stage where you do a stage 1 clinical trial, a phase 1 clinical trial. And in this phase 1 clinical trial, you're really not even testing for efficacy. You're not testing to see if the vaccine is actually working. What you're testing for mainly is safety. So there are two major things that you test for in a vaccine. You test for safety and you test for efficacy. Safety is the most important component because if the vaccine is not safe, you cannot go any further. Why? Because unlike other drugs, vaccines are given to healthy people. And you don't want to give something to a healthy person that causes problems in that healthy person. As a result, if you actually get approval to go into phase 1, it's a small number of people, typically on the order of 10 to 50 people, but no more than 100, because you don't know what sort of side effects and responses you're going to get. If you've actually demonstrated safety in phase 1, then you can go to a phase 2 clinical trial, where you actually get more people. You stick with a particular dose because now you would have a very good idea of what dosing to use, because you would have broken up people and stick phase 1 into those different groupings. And now you have a few hundred people. You're seeing how that goes in phase 2. Is it safe? And number 2, do you have a good dosing? Are you getting immune response? And then if you're actually getting the type of response that you want, you can go into phase 3 clinical trial, which is a large clinical trial, tens of thousands of people, where essentially what you're looking for is actually to have a group of people who are on a placebo, those who do not get the vaccination, and those who get the vaccines. And it's double blind, so neither the patient nor the physicians will know. You have an independent company that randomizes people and give this to them. And you also want the pandemic or the disease to be continuing. So for coronavirus, if all of a sudden the virus goes away, you won't be able to do a phase 3. Because what you want is that you want people to be exposed to the bug to see who gets infected and who doesn't. And after that, you still have to continue with production and efficacy and all of that. But what you're looking for essentially is that you're looking for neutralizing antibodies that will bind to the virus and neutralize it and prevent an infection. And this is really just showing you all the different versions of a vaccine that could be made against coronavirus that I talked about. And what has gotten a lot of attention is this RNA version of the vaccine, where essentially you could put in the messenger RNA. This is what Moderna is working on. And you kind of bypass all of this protein issues that I was talking about that I had to do with my nanoparticle, express these proteins and make sure that they're expressed. Because the messenger RNA, when it gets picked up through the lipid molecules that it's embedded in by the cell, it immediately gets into the cytoplasm of the cell, gets in the ribosome, and you have the proteins being expressed. And the expression of that specific protein now leaves the immune system to make antibodies against it. And that now can give you a good protection. But I have to caution and agree with my friend Dr. Osterholm, who says that you can make a vaccine against anything very quickly, but it needs to be effective and it needs to be safe. And this is a long process and can take months to years to develop, as I showed you. And so as you remember, RNA is this DNA intermediate that you have the DNA in the nucleus. You have a copy of that DNA that now goes into the cytoplasm, so you can actually have translation. So there's a transcription for it, so you can have translation into the protein. And so Moderna is one of those companies that's trying to make one of these vaccines. But again, as I said, you can't skip these periods because let's look at timelines here. You know, typically it takes about 5 to 10 years, like me, over 10 years now in the preclinical testing, before you actually get to clinical trials. And if everything is going well, you can have maybe 3 years per phase of the clinical trial. What they're doing now is that they're actually accelerating the SARS-CoV-2 vaccine production. Warp speed is actually blending these areas, so you can see that there's overlap between the clinical testing and phase 1 and between phase 1 and phase 2. So Moderna is in a phase 1, phase 2 at this stage. And the estimate is that they're thinking that they can actually make a vaccine within a year to 18 months. That's assuming that nothing at all goes wrong. And so right now we have over 130-something vaccines, close to 140 vaccines in clinical trials, in development, sorry. 125 of them are still in that animal testing, lab testing stage. 7 of them are in phase 1, another 7 in phase 2, and mainly it's because phase 1 and phase 2 have been overlapping. And then people are talking about a phase 3 now with the Moderna vaccine that they're expecting to go into a phase 3 clinical trial with starting at the end of the summer. And so essentially, as I wrap up, what people are expecting is this overlapping. So this is what I told you about regular vaccine development and licensing. Here's what is happening with SARS-CoV-2 vaccine or COVID-19 vaccine, where preclinical trials are overlapping with safety trials, are overlapping with production. So typically the production phase of a vaccine could take years. What they're doing now with the Moderna vaccine and a few others is that once it clears phase 1 clinical trial into phase 2, they're actually starting upscaling of manufacturing so that if it works, you'll have a vaccine quickly. If it doesn't work, you'd have spent billions of dollars on something that you have to throw away. And so essentially the government has pumped a lot of money into this part of it, the manufacturing part of it. But we learned some lessons in 1918 from the flu pandemic that will probably be good for us to remember. So this is from Morgan Stanley, their projections of what is happening with this outbreak. So we're somewhere right around here, where what we've been seeing is kind of a reduction in overall rates in the United States. For some places, they're seeing a little bit of spike. We're doing your regular RNA testing, PCR, and then antibody testing. And the thought is that maybe as the summer comes on, the levels of infection will go down, but the expectation is that there'll be a second wave. In 1918, we had a second and a third wave. And unlike this projection here, the second wave for the 1918 flu pandemic was caused 5 times more death than the first wave. But essentially, the thought is that there might be another wave. And the projection is that we potentially wouldn't have a vaccine to start distributing until about March, April of 2021, if that early. So the lifestyle that we have now, we have to get a little bit used to it, because the probability of having a vaccine at this point is going to be less. So finally, I just want to acknowledge the students in my lab who did the work. Tawana Childs worked mostly on the gas vesicle production, and between Sam and Melissa and Dorothy and Mina did the animal work that led us to the point where we are. And I thank you very much for your attention.
Thank you so much for your presentation, Dr. Webley. The Q&A session will be moderated by Leah and Karen. Go ahead with questions.
Okay, thank you, Dr. Webley. Our first question comes from the audience and saying, with antibiotics, evolution can prompt bacteria to develop resistance to those antibiotics over time. Are vaccines also prone to being rendered ineffective through viral mutation?
Very good question. Absolutely. We see it with influenza. That's why we have to get a new influenza vaccine every season. With SARS-CoV-2, though, it has something that a lot of RNA viruses does not have. It actually has a proofreading machinery. So, you know, as nucleotides are added, as you remember, viruses like HIV make a lot of mistakes because it doesn't have a good polymerase. The polymerase doesn't really care. It just adds stuff in. But SARS-CoV-2 actually has a very good proofreading system. So what that means is that it doesn't make a lot of mistakes, which means that the probability that your vaccine is not going to be effective because the virus has mutated is very low. So at this stage, we really haven't seen a mutant form of the original virus, even though we're seeing variants. The variation is not enough for you to have the same antibodies not responding. So that's good news for this virus.
Okay. Outside of the current pandemic and a vaccine for COVID-19, what would you say was the most important vaccine for a disease that currently does not have one?
For a pathogen? Essentially, what vaccine do we need to develop now that we don't have?
Yes.
Yeah, I mean, of course, I'm going to be a little bit biased, and I'm going to say sexually transmitted diseases like chlamydia and gonorrhea. We definitely need vaccines for those. It was really great to have an HPV vaccine in 2006, 2007, because that was also a really problematic disease. And the reason why I say chlamydia is because of just the sheer numbers of people who have this disease and the mortality and the morbidity mainly that we see from this. Infertility is a major problem worldwide. And one of the greatest reasons for infertility is the damage that is caused by chlamydia organisms. So I would say this is an important one at this stage. Of course, we're looking for, we just got a vaccine to Ebola, and we're looking for vaccines to other types of viruses like this that seems to be a big problem.
Okay. Has a phase 3 trial ever been interrupted or canceled because a pandemic stopped naturally?
Yes. That happened actually with the first SARS virus. So it went away so quickly that people who were developing these vaccines didn't get a chance to do it. And I think it might have happened with Ebola. initially. And so people never made an Ebola vaccine. So we didn't have an Ebola vaccine until last year because of that reason. And so, it's not an unusual thing for the pathogen to just disappear over time and you don't get to complete vaccine development.
Okay, so one attendee was reading an interview with Dr. Alsterholm, who stated that they weren't successful in creating a vaccine for either SARS or MERS. And what was so different about these that they neither killed anywhere near as many people or that they naturally sort of went away? And do we think that this will be similar in COVID-19? Is it a possibility?
Yeah, my wish is that it just goes away. But as an infectious disease person, I know it won't. The major differences with this virus compared to SARS and MERS, number one, just how contagious it is. This virus is way more contagious than anybody initially thought. And not only is it contagious, the amount of organ systems that it's able to impact is very, very different from the pathology that we saw with SARS or even MERS. Even though those viruses killed more people, it wasn't as contagious. In other words, the affinity that SARS-CoV-2 has for the ACE2 receptor far, far outweighs that of the MERS virus. So because of the affinity that it has for its receptor, it binds much more tightly, so it doesn't let go and it gets into the body much more rapidly. It probably is also that people who had MERS and SARS get so sick that that they're sick, so they don't get to transmit the virus silently the way that we've seen this virus do it. And so although the WHO kind of messed up this week in talking about the transmissibility of the virus, what everybody who works with these patients will tell you is that this virus can readily be transmitted from people who are showing no symptoms. Whether it is that they're going to be asymptomatic throughout the entire period or that they are pre-symptomatic and would have symptoms later, they can transmit the virus. And because of that, you're having this massive transmission of the virus and the type of pathology that we're seeing.
And unfortunately, that's all we have time for questions. Thank you so much, Dr. Webley, for that enlightening and kind of scary presentation, and for all the effort that went into pulling your slides together. Those were really wonderful images.